we're going to discuss sporulating and toxigenic bacteria. The objectives for this lecture are listed here. I'm not going to go through them item by item, but they will provide a framework for you to prepare for the material when the time comes for assessment. Okay. So here first, we'll conduct an overview of bacterial exotoxins. Many bacterial diseases are caused by exotoxins. For example, well-known bacterial diseases such as cholera and tetanus are caused by exotoxins, which are diffusible proteins with enzymatic properties. Exotoxins are produced by specific strains that are implicated in the disease. There are non-virulent strains of the same species that do not produce the toxin and do not cause the disease. Exotoxins are produced during exponential growth phase. Both gram-positive and gram-negative bacteria can produce exotoxins. Exotoxins can be encoded, this is important, they can be encoded on the bacterial chromosome, on the pathogenicity islands on the chromosome, or on resident plasmids, resident phages, or transposons. So the genes for exotoxins can be anywhere within a bacterial cell. Okay. In this slide, we'll look at various definitions: exotoxins, toxoids, endotoxins, virulence factors. Our focus for the remainder of the lecture is the highlighted one, exotoxins. Exotoxins are usually proteins or polypeptides. They usually act distally from the infection site, but can also act locally at the infection site, and some cases function as invasions. Okay? They usually have enzymatic activity, but some exotoxins are superantigens. We'll discuss those briefly. As contrasted to exotoxins, toxoids are simply denatured forms of exotoxins. They have no toxicity, but they do retain antigenicity. In fact, they are very good vaccines. Endotoxins, on the other hand, are cell-associated molecules, usually lipopolysaccharides in gram-negative cell, and they usually cause an inflammatory response. We're not going to consider endotoxins in this lecture. Virulence factors are those that are necessary for certain bacteria to invade into tissues, and these factors are usually injected into host cells, okay, into specific target host cells. There are some similarities between virulence factors and exotoxin, but with the only topic we're going to discuss here are exotoxins. Okay. There are three basic types of exotoxins: the A-B toxin, the membrane disrupting toxin, and superantigen. These are the three major types of exotoxin. Within these three, the most important group is the AB toxin. In AB toxin, there is an invariant overall structure. There are two subunits. One is the A subunit, the other is the B subunit. The A subunit is the enzyme. It's also called the active subunit. Okay? The B subunit is the receptor binding subunit. It usually is combined with another domain called the translocase domain that has a specific function that we'll discuss shortly. So A is the activity or enzymatic activity. B is for binding to the cell, the receptor binding. Okay? So there are subclasses of AB toxins. The simplest is simply one unit of A, one unit of B. Okay? Somewhat more complex examples are A5B. There is one subunit of the enzymatic activity and 5 molecules of the binding domain B. This is an A5 kind of, A5B kind of complex. There's another kind of complex, A2B, where there are two different A activities on sharing one B binding domain. We'll see examples of these in subsequent slides. Okay? There are two types of membrane disrupting exotoxin. One type is a passive channel. So this exotoxin simply inserts itself into the membrane of a host cell and creates a channel. You might want to call it an illegal portal that will allow the escape of ions into the environment and the influx of water into the cell. Right? So they're passive pore-forming toxins. There's another kind of membrane disrupting exotoxin that's a phospholipase. That's an enzyme that actually chews away a component of the cell membrane, host cell membrane, to disrupt the cell membrane, again with the same result. That is, um, um, that is a outpouring of ions and an inpouring of water, ultimately leading to killing of the cell. Right? A superantigen, on the other hand, is does not have enzymatic activity, but it has the ability to provoke a very strong and often lethal inflammatory response. Right? We'll see what that means in a subsequent slide. All right. Let's first look at a generalized mechanism of how AB toxins work. This generalized mechanism is shown in the black and white diagram on the left side. A variation of the same mechanism is shown in the colored diagram on the right side. Please focus on the black and white diagram on the left side first. The AB toxin is usually synthesized by the bacterium in an inactive form called a toxinogen. The toxinogen must be cleaved to create an active AB toxin. Okay? In this case, the C domain keeps the toxin from acting. Proteolytic cleavage of C releases AB, which is the active toxin. The AB active toxin then binds to a specific cell surface receptor of the host target cell, let's say the human cell. Okay? Specific receptor is recognized by the B domain. Once recognition happens and the toxin binds to the host cell surface, then the active subunit A is internalized and it is released into the interior of the cell where it carries out its enzymatic function, which ultimately has a toxic effect, most usually killing of the cell. Okay, ultimately killing of the cell. Okay, so the AB toxin binds to the surface of the target cell using the B domain. The active domain A is internalized and allowed to exert its enzymatic function. This is the generalized mechanism of action of an AB toxin. A slight variation of that is shown in this diagram at the top. Pretty much the same as what you see here, except that after the B domain binds to the surface, the entire toxin is endocytosed into its own vesicle and is delivered to the interior. From that endosome, the A unit is released into the cytoplasm. So ultimately, same thing as what's happening here, except that in the first step, instead of B binding to the surface and the A becoming internalized into the cytoplasm, there is an intermediary step that is an endosome. From there, the A subunit is internalized into the cytoplasm. So that's the generalized mechanism of how AB toxins work. All right, here's an example of an AB toxin, the diphtheria toxin shown in a ribbon diagram. The red part is the A subunit, the green and blue together is the B subunit. The B subunit, the green part is the receptor binding domain. The blue part is the translocase function of the B subunit. Okay, so this AB toxin is synthesized as a single polypeptide, but before it can act, A is cleaved from the B domain. However, they are still covalently held together by a disulfide bond. So there is no peptide bond between A and B, but there's a disulfide bond. Okay? So A is internalized via B-mediated endocytosis, and subsequent translocase action moves the A subunit into the cytoplasm. The function of A, enzymatic function of A, is to ADP ribosylate elongation factor 2. That is the translation elongation factor 2 to block translation. What ADP ribosylation means is shown in the next diagram. Okay, first, here is an example. This is a compilation, not an exhaustive compilation, of different AB toxins and their enzymatic activity and their biological effects. You don't need to memorize this, but we're going to see some of the toxins in greater detail anyway in subsequent slides. So the diphtheria toxin, we're going to consider that later. Shiga toxin, we're not going to consider that. Pertussis toxin, yes, we're going to consider that. Cholera toxin, no, we're not going to consider that. Botulinum and tetanus, we're going to discuss them both. What you can see here, there's different kinds of enzymatic activity associated with AB toxin. For example, ADP ribosylation of elongation factor 2. Shiga toxin can do something different. Cleavage of ribosomal RNA. Botulinum toxin does something else. Cleavage of a specific protein involved in signaling, so synaptobremin. Okay? So the consequences are also various. For example, diphtheria toxin blocks protein synthesis. Botulinum toxin causes a kind of paralysis called flaccid paralysis, so on and so forth. Okay, we'll see specific examples of mechanisms in subsequent slides. Okay, first, let us take a look at enzymatic activity, some enzymatic activities of AB toxin. One of the most common enzymatic activity of AB toxins is the so-called ADP ribosyl transferase activity. ADP ribosyl transferase activity transfers ADP ribosyl from NAD. NAD structure is shown here. It consists of adenine, ribose, 2 phosphates, another ribose, and nicotinamide. Okay? So ART activity acts on NAD and transfer this entity, ribose, 2-phosphate ribose, and adenine, to a lysine residue in a target protein. This is the so-called ADP ribosylation reaction. A single moiety is transferred to a single lysine residue in a target protein, and that transfer is sufficient to modify the function of the target protein. Okay? For example, inactivation of elongation factor 2 by diphtheria toxin falls into that category. There are other host enzymes also that are targeted and inactivated by this mechanism. Some A subunits, however, don't have ADP ribosyl transferase activity at all. Instead, they have a peptidase activity. They are proteases or endopeptidase activity. For example, tetanus, anthrax, and botulinum, all of these have endopeptidase activity. Some A subunits have a different kind of activity, adenyl cyclase activity. Adenyl cyclase is an enzyme that converts AMP to cyclic AMP. Okay? For example, one of the anthrax toxins and a toxin produced by Bordetella pertussis have this kind of enzymatic activity, which is adenyl cyclase activity. Okay. We discussed that there are two types of membrane disrupting exotoxins. One is the kind that introduces a porin or a pore into the cell membrane. This action is shown in this cartoon form here. Here the toxin basically shown here by the two black cylinders inserts itself into the host cell membrane, creates an illegal channel that allows ions to diffuse out and water to diffuse in, leading to cell swelling, and ultimately lysis. There are examples of this kind of toxin or Clostridium perfringens toxin, enterotoxin. We're going to come to that in one of the final slides. Another example is uropathogenic Escherichia coli RTX toxin. We're not going to discuss that particular toxin further. We're going to discuss Clostridium perfringens toxin though. Okay. The other kind of membrane disrupting exotoxin is actually a phospholipase. Here, the enzyme basically chews away lecithin, which is part of the cell membrane, destabilizing that area of the membrane, again with the same result. That is an outpouring of ions and then an uptake of water leading to cell lysis, cell killing. An example of that is a different toxin produced by Clostridium perfringens called alpha toxin. This is an important toxin. Okay. Finally, we come to the mechanism of action of the third kind of exotoxin. This is the so-called superantigen. To refresh your memory, a superantigen is a kind of exotoxin that has no enzymatic activity, but it has the property of provoking very strong and sometimes lethal inflammatory response. How does the superantigen work? That is shown diagrammatically on the left side, and the same point is made in a somewhat more elaborate diagram on the right side. An example of a superantigen is a exotoxin produced by Staphylococcus aureus called TSST1, that stands for toxic shock syndrome toxin 1, TSST1. So to understand a superantigen, we have to first look at what a normal antigen is and how a normal antigen provokes the immune system, how it modulates the immune system. The normal situation is shown on the left-hand panel. When an antigen is encountered by the immune system, the antigen is taken up by a type of immune cell called the antigen presenting cell. There are many antigen presenting cells, but basically they all do the same thing. They pick up a component, antigen, a component of a bacterium, process it a little bit, and present it at the cell surface in a form that is bound. In this case, the antigen that's being presented by this antigen presenting cell is shown as an oval, green oval here. And the green oval is held at the surface by a surface molecule that belongs to the APC, antigen presenting cell, called the MHC class 2 molecule. So the function of MHC class 2 molecule is to hold the processed antigen at the surface so that that antigen can be recognized by T cell. The T cell is shown at the top. Now, depending on the antigen, a small subset of the T cell, in fact, a very small subset, 0.1% of the entire T cell population recognizes each type of antigen. Okay? And the recognition occurs between is T cell receptor, shown as TCR here, which recognizes in a specific way the antigen presented by the antigen presenting cells. As a result of this interaction, T cells are activated. Ultimately, these T cells will activate B cells, which then ultimately lead to production of antibodies that target this particular antigen. The bottom line here is that each antigen can only activate a small fraction of the T cell population that is present in the host organism. So how does this situation differ from a superantigen? If a superantigen is present, what happens to the immune system? Something very drastic happens to the immune system. It turns out that whereas this interaction of this antigen is very specific and limited to only a small subset of T cells. The presence of superantigen, which is shown here by the green oval, this color scheme is unfortunate because the same color shown here. Anyway, the superantigen, which is shown as the green oval, acts as a bridge that glues any antigen presenting cell, regardless of what antigen is held by the APC cell, to a T cell. The end result is, instead of a small subset of specific T cells reacting with an antigen, regardless of what the antigen is, that recognition between T cell and APC are promoted by superantigen, such that a very large fraction of the T cells are stimulated, and as a result, there's a massive immune response and leading to something like toxic shock syndrome. Right? That's the way the superantigen functions. The same points are made here. Here is the T cell receptor, and here is the MHC class 2 molecule on the surface of antigen presenting cell. Here is an antigen, any antigen, it doesn't matter. Regardless of what the antigen is, a T cell is forced to recognize it because of this bridge formed by the superantigen that leads to a massive provocation of T cell and a massive immune response that has lethal consequences. Okay. Let's go on now to a list of the bacteria that we're going to be looking at and their staining properties, their metabolic properties, whether they form spores or not, and what diseases they cause. Okay, so we're going to look at Bacillus anthracis, Corynebacterium diphtheriae, Bordetella pertussis, Pseudomonas aeruginosa, Clostridioides difficile, Clostridium tetani, botulinum, and perfringens. Okay. I'm not going to go through the entire table, but it's here for your quick reference. Okay, let's start with Bacillus anthracis and anthrax toxin. There are three types of anthrax intoxications, and those are cutaneous, inhalation, anthrax intoxication, and gastrointestinal anthrax. Right? All right, first, a little bit about the organism. No? These are large gram-positive aerobic rods with square ends. Right? You see in the micrograph, the ends are square. Right? These are encapsulated. Encapsulation in general means they cannot be phagocytosed easily by the host immune cell. Right? They have a centrally located spore, which becomes evident upon nutritional depletion. Spores that are in the center of the microbial cell. Okay? The Bacillus anthracis was the first bacterium shown to be the cause of a specific disease, and that was shown in 1877 by Robert Koch, who actually grew the organism in pure culture, demonstrated its ability to form endospores and produce experimental anthrax by injecting into animals. You might remember something called the Koch postulate. This was the origin of the Koch postulates. Okay. The epidemiology of anthrax is as follows. It is basically an animal disease that occasionally intrudes into the human population. It typically grows in sick animals, especially herbivores such as cattle, sheep, etc. So the spores produced by the organism remain in the soil for a very long time. The disease is distributed globally, but it's much more common in developing countries. In the United States and in Western countries, because of livestock vaccination, anthrax is not very common. Right? This disease, there's no human-to-human transmission. This is an intoxication, not an infectious disease per se, except under specific conditions that I'll refer to shortly. Okay? The disease is controlled mostly by proper animal care and proper disposal of dead animals, but the disease probably cannot be eliminated because it's a disease of herbivores. Okay. There are three forms of anthrax that are referred to. The most common is the cutaneous. The least common is gastrointestinal. The cutaneous form is the situation where spores germinate typically in something like an abrasion or a cut in the skin, and they produce toxin locally, which then has activity that is often lethal. The inhaled form of anthrax is what happens when you inhale spores that germinate in the lungs and produce toxin. This is the next most common. This is also the form of anthrax that has been weaponized as a biological weapon. And the least common is the gastrointestinal form, where you ingest food that has spores. The spores survive the gastric acid and germinate in the intestine, and they can produce the toxin, which is then absorbed and leads to systemic intoxication. Okay, cutaneous anthrax. As we mentioned, spores enter the site of an abrasion or a cut, and that leads to their germination, and the symptoms are itching and a lesion that ultimately turns black. It's called a black eschar. Okay? It can be fatal, but it responds well to antibiotics. And the bacteria typically do not invade. They do not cause systemic infection. They remain local. Inhalation anthrax, on the other hand, is sometimes called wool sorter's disease because it was common in people who handle sheep, wool workers. Okay? Inhalation of spores leads to the disease because of germination of spores in the lung. The symptoms are very similar to common cold, but they can abruptly change to very severe symptoms, including fever and shock. And the disease is often fatal. And in order to trigger inhalation anthrax, you need to inhale 8 to 10,000 spores, substantial exposure. Mhm. Now, okay, the GI anthrax, which is the least common form of anthrax, is because of ingestion of spores in contaminated meat. And here the symptoms are nausea, loss of appetite, vomiting, fever, abdominal pain, vomiting of blood, severe diarrhea, things like that. And it also can be fatal with a 25 to 60% mortality. Okay. Mhm. So the virulence factors of anthrax, there are three of them. One is actually a, a capsule, polygamma D-glutamate capsule, and the genes encoding the glutamate capsule are carried on a large plasmid called PXO2. There are two toxins that are important. Both of them are encoded on a separate large plasmid called PXO1. And spore formation, that is, ability of the spore to survive adverse environmental conditions, is also considered to be a virulence factor of bacillus anthracis. Okay. The bacillus anthracis capsule is correlated very well with the disease. That is, strains that have the capsule or the pathogenic strains. Okay? And as we mentioned before, the function of capsule is protection against phagocytosis. Okay? And the strains of Bacillus anthracis that do not cause disease typically do not have the capsule. Good. Anthrax toxins is an AB type of toxin. It's a complex AB toxin with two different A subunits and a single B subunit. The B component also has the special name protective antigen or PA. So PA and B are the same thing. It binds to the cell receptor and facilitates the transport of the two different A subunits. The two different A subunits are called EF and LF. A component 1 is called EF for edema factor. This is an adenylate cyclase. That is, this is an enzyme that converts AMP to cyclic AMP. The A subunit 2 is called lethal factor. This is a protease. This is a zinc-dependent protease that cleaves a specific kinase called MAPK kinase that ultimately leads to cell death. Mhm. The anthrax toxin was recognized in 1954, and serum containing toxin caused anthrax symptoms when injected into guinea pigs. That's how we know that it is this toxin that causes the disease of anthrax. Okay. In this slide, we summarize the various enzymatic functions of the anthrax toxins. To refresh your memory, PA is the B subunit. It has a special name, protective antigen. Okay? The two A subunits are called EF and LF. EF for edema factor, LF for lethal factor. EF is an adenylate cyclase, LF is an endopeptidase. Here are the mechanism of action of these toxins. The anthrax toxin, which is an A2B toxin, initially binds to the cell surface at specific receptors in the diagram shown by this dark pink molecules at the surface. These are the receptors to which the PA heptamer binds. The B subunit heptamer binds, and EF and LF bind to this heptamer and they are internalized into the cytoplasm. Okay? Now, the EF or edema factor is shown in blue and lethal factor, LF, is shown in green. Unfortunately, these two colors are not very well differentiated, but there is that color difference. This is blue, this is green, okay? Blue is edema factor, green is lethal factor, okay? Both of them are delivered to the interior by binding to the PA heptamer at the cell surface. Edema factor basically associates with the cell, the host factor called calmodulin. Together, they form an adenyl cyclase that results in an increase in CAMP levels inside the cell. CAMP elevation in general disrupts cell signaling, ultimately leading to apoptosis. Apoptosis is a programmed cell death of a host cell. Right? The lethal factor, on the other hand, which is also delivered via binding to the PA heptamer, has an endopeptidase activity that targets one specific protein. That protein is MAPK kinase, which is a part of the signal transducing cascade in the cytoplasm, again leading to the same consequence, which is cell death. So both of the toxins ultimately kill the cell. Okay. Bacillus anthracis spore formation proceeds through distinct, well understood stages. In conditions of starvation, one of the bacterial cells, one of the daughter cells produced by cell division, is internalized by the larger cell, and that internalized daughter cell ultimately becomes the spore. That spore is released into the environment. The spore has very thick cell walls as well as other protective mechanisms that allows it to survive harsh conditions. Then when more amenable conditions become available, it germinates to form a normal Bacillus anthracis cell. A testament to how tough these spores are is that they're actually resistant to heat, drying. They're also resistant to 95% alcohol. Very few living things are resistant to 95% alcohol, but these spores are. Okay. As you all know, anthrax has been developed um, uh, as a as a biological weapon. Okay? It's not currently actively developed, but it was developed as a biological weapon. It was developed first by the British Army. They tested it for the first time in 1942, and it was subsequently also developed and tested both in the Soviet Union and the United States. However, in the late 1960s, uh, all research on anthrax weaponization was ended by a presidential order, and the building where the research was conducted was actually demolished subsequently. Nevertheless, you might remember or you might have read about an anthrax attack that was mounted in 2001, and this was an attack that was never really solved. It's not known who actually launched that attack. There are lots of theories about it, but ultimately there's no resolution to who launched the attack. Only a very few people could have launched the attack, but who did that is not established. Anyway, here is what the issue is. To weaponize anthrax spores, you need the ability to disperse the spores very widely. That is, although anthrax toxin is extremely potent, you need only a few molecules to kill a human being. It's not easy to disperse them because the spores stick together because of van der Waals forces. However, people discovered early on in the 1940s, the British and the Soviets and the United States, they all discovered that if you coat the spores with silica, then the clumping does not occur and the spores remain discrete and they're much easier to disperse. This was basically what was achieved in weaponization. And somebody in the 2001 attack knew how to do it. It's extraordinarily difficult to achieve that without killing yourself, but somehow it was done. And the anthrax attacks are well documented, but the case is still not closed. Okay. Anthrax is diagnosed on the basis of gram-positive staining, the presence of spore-forming rods, and so on. The treatment, antibiotics can be used for respiratory and GI anthrax, and but it doesn't prevent mortality sufficiently because by the time you discover an infection, a toxin has been made already. Monoclonal antibodies against protective antigens was recently approved for inhalation anthrax. However, effectivity is still not known. Anthrax is not a very common disease, obviously. Yeah. All right, now we move on to the next toxigenic bacterium, which is Corynebacterium diphtheria. Corynebacterium diphtheria toxin is also an AB toxin. First, a few words about the bacterium. It's a gram-positive bacillus. It's pleomorphic, slender, and it has a clubbed ends and palisading. Whenever I show a morphology in red, that means that I'm drawing your attention to it. Clubbed ends and palisading means stacking. These features are important about Corynebacterium. It's also aerobic. It causes, obviously, an infection of the throat and the mouth, which are oxygen-rich environments. Okay? To grow Corynebacterium in the laboratory for diagnostic purposes, you use a kind of agar called tellurite agar. Tellurite agar has a salt of the metal tellurium, and this is an example of a selective agar. When you streak a sample, a swab from a mouth onto tellurite agar, most bacteria cannot form colonies, but Corynebacterium can form colonies, and those colonies are black in color because Corynebacterium can reduce the tellurite salt into the metallic tellurium, which is black in color. So this bacterium colonizes throat and the nasopharynx. It's not invasive, stays local, but it forms a thick gray mucoid coat shown in this patient's throat. Okay? And it's also called a pseudomembrane that, that when scraped in the doctor's office leaves a bloody patch when it's removed. Okay? The symptoms of diphtheria are sore throat, fever, cough, lymph node involvement, and there are complications of infection on heart, peripheral nerves, kidney, and so on. Okay? Diphtheria toxin is a classic AB toxin. Diphtheria toxin production is regulated by iron levels in the environment that the bacterium finds itself. When the iron levels are low, the toxin is produced at high levels. Right? And the AB toxin binds to a particular receptor called the HBEGF receptor. This receptor is present in virtually all human cells. Okay? And the B domain binds to that particular receptor and internalizes the A domain. The A domain, ADP ribosylase elongation factor 2, which blocks translation, ultimately killing the cell. Okay. Here's the incidence, treatment, and prevention of Corynebacterium diphtheria. The incidence is very low in the United States currently. It used to be 200 per 100,000 in the 1920s. Today, it is extremely low, 0.001 per 100,000. That's because of vaccines. The mortality is about 10% for infected individuals. It's higher in very young children and adults older than 40. The worldwide, there are 5,000 deaths. Mostly, almost all of them are due to lack of vaccination. Treatment for the disease is mostly by antitoxin to neutralize the diphtheria toxin and antibiotics to kill the bacteria. Prevention is by vaccination, DTaP and TDAP combination vaccine, which are given together for diphtheria, tetanus, and pertussis. Mhm. All of these vaccines are made by toxoid, which is denatured toxins. Mhm. Next organism is Bordetella pertussis, and this is the organism that makes the pertussis toxin, as well as a second toxin, which is called a bacterial adenyl cyclase toxin. Okay. The organism is described as a gram-negative coxobacillus, which is also described as a dumpy rod. It's a fastidious, slow-growing organism. Fastidious means demanding in what it needs in the media to grow. It colonizes the region between airways and lung, so obviously this is an aerobic organism as well. The symptoms are like a cold symptoms, but it's characterized by a characteristic spastic cough called whooping cough. Okay? It's an extremely contagious disease and is a significant cause of morbidity worldwide. About 16 million cases and about 200,000 deaths annually worldwide. However, in the United States, the incidence is very low, again, because of vaccination. So the Bordetella has an important virulence factor called filamentous hemagglutinin, but it has two exotoxins. One is adenyl cyclase, also known as bacterial adenyl cyclase, and another toxin called the pertussis toxin. It turns out that both bacterial adenyl cyclase as well as pertussis toxin have ultimately the same effect inside the host cell, and that effect is elevating cyclic AMP or CAMP levels through two different mechanisms. So they both achieve the same result, elevation of internal CAMP levels, which in turn triggers apoptosis and cell death. Okay. How they do that is shown in the diagram, which appears to be complicated, but I promise to walk you through to show you that the basic ideas are fairly simple. we said that there are two different toxins. One toxin is a bacterial adenyl cyclase. And adenyl cyclase is an enzyme that converts convert AMP to CAMP, right? And the function of the bacterial adenyl cyclase is shown in this part of the diagram. The adenyl cyclase toxin is shown here, which consists of a binding domain shown in green here and an activity domain shown in yellow here. And the binding domain binds to the surface of the target cell, target whole cell, and the yellow domain is internalized, which then associates with a cell protein called calmodulin to create the adenyl cyclase that then converts AMP to CAMP. CAMP levels go up. So this is very simple. It's a classic AB toxin that promotes CAMP formation. Right? Now, the same result, that is elevation of CAMP, is also achieved by the pertussis toxin, which follows a more complicated route to achieve CAMP elevation, and that route is shown on this side of the diagram. Pertussis toxin acts by blocking an inhibitory pathway whose function it is to keep CAMP levels low. Now, when CAMP levels are high, the cell goes into cell death pathway called apoptosis. Therefore, the CAMP levels need to be kept low. And that is achieved by a signal transduction pathway whose ultimate effect is to inhibit the formation of CAMP. Pertussis toxin has the effect of compromising that signal transduction pathway so that the inhibition of CAMP production is blocked. How it does so is shown in the diagram here and this portion of this diagram. What is shown here is the cell membrane of the host cell, and is shown a specific receptor called GPCR, which stands for G protein coupled receptor. This is a very common kind of cell receptor that receives a variety of signals at the cell surface. When it receives the signal at the cell surface, it leads to activation of an associated proteins called G proteins that are in the internal face of the cell membrane. It's a G proteins, right? Now, when a signal is received at this receptor, the G protein is dissociated and that acts to inhibit CAMP protection. What pertussis toxin does is it modifies the G protein such as to interrupt the pathway. So the pathway is interrupted, therefore CAMP production continues without interruption. So both the adenyl cyclase pathway and the pertussis toxin pathway ultimately lead to a runaway production of cyclic AMP, and therefore they trigger cell death by apoptosis. All right. Treatment for bordetella pertussis is through antibiotics. It only changes course of the disease, but it's only effective if it's given early. Bordetella pertussis is actually a very serious disease, and sometimes it requires adequate oxygen support to keep the patient alive. Prevention is easy again, the same combination of vaccine DTaP is effective in keeping bordetella pertussis at bay. Okay, Pseudomonas aeruginosa, the next organism, is actually an environmental bacterium. This bacterium only affects people with some sort of a compromise, immune compromise, a patient that have catheter, or patients who have cystic fibrosis and such. They produce many different toxins. Two we're going to consider here are exoenzyme S and exotoxin A. Okay. First, a little bit about the organism. It's a gram-negative bacillus. It's a soil organism found in soil, water, animals, and so on. It has some cultural characteristics, quite unique characteristic. Colonies for Pseudomonas aeruginosa have a fruity odor, sometimes described as an apple-like odor, and they form a blue-green pigment on a particular kind of agar called a trypticase soy agar. Blue-green pigment is produced by the bacterium. So the pigment comes from the bacterium. It produces this pigment that appears green. And Pseudomonas aeruginosa has almost no nutritional requirement, meaning that it's a good example of a prototrophic organism, an organism that synthesizes pretty much everything it needs from very simple ingredients. It's an aerobic organism, and it cannot carry out fermentation. So as we mentioned, Pseudomonas aeruginosa is strictly an opportunistic pathogen. It only compromises immune compromised patients or patients with catheters, etc. It can cause both localized infection, like cystic fibrosis patients, lungs of cystic fibrosis patients, and can also cause systemic infections. Okay? Some examples of situations with Pseudomonas infects patients is given here. The virulence factors for Pseudomonas aeruginosa are alginate, which is like a thick gel, which is also has the same function as other similar protective envelopes, which it say is anti-phagocytic. It produces two major toxins, produces other toxins as well. Two we're going to consider are exoenzyme S, which is an ADP ribosylation, which is a ADP ribosylase. It also affects G proteins, different G proteins than the ones we considered in a previous organism. The second toxin is exotoxin A, which ADP is a ribosylase elongation factor 2. Most Pseudomonas aeruginosa strains carry exotoxin A. Only a few carry the gene for exoenzyme S. Pseudomonas aeruginosa treatment requires very aggressive use of antibiotics, very high doses, because it's a very resistant organism. And prevention is mostly by paying strict attention to control measures, because Pseudomonas aeruginosa infections are most common in hospitals. Treatment, as we mentioned, is by using aggressive use of at least 2 antibiotics at very high doses. And okay, so much about Pseudomonas. We move on to Clastidiaedes difficile, which we encountered already in a previous lecture. Now, C. diff is the organism that leads to the symptom called pseudomembranous colitis, and this is because of toxin called TCDA and TCDB. In this lecture, we're focusing on the toxin, so we'll look at what TCDA and TCDB are. The organism itself is a gram-positive organism. It's a strict anaerobe, and we learned all of this stuff in the previous lecture, so I'm going to go quickly to the actual toxin. The C. diff toxin is an AB toxin. It acts by glucosylating small G proteins belonging to the Rho family. Okay? When they glucosylate Rho family GTPases, G proteins, the immediate result is actin condensation. Actin is part of the cell cytoskeleton. By acting on the cytoskeleton, they lead to a condensation of the actin, and which in turn triggers apoptosis and cell death. So this is an AB toxin that ADP ribosylase Rho family G proteins, ultimately triggering apoptosis. TCDA and TCDB are two different AB toxins, but both have the same functional effect. They're in fact actually homologous, and they're also homologous in their function. They otherwise are classic examples of AB toxin. They are endocytosed, and then the active domain is released. The active domain has the enzymatic characteristic that triggers cell death. Okay. Now, pseudomembranous colitis, about which you learned a little bit already, basically there's no new information here. This information is the same as what we discussed in the previous lecture. I'm going to skip over that and go into the next group of organisms, and those are all clostridial pathogen. Clostridium tetani, Clostridium botulinum, Clostridium perfringens. Okay? And the toxin produced by these organisms are tetanus neurotoxin, botulinum neurotoxin, and the clostridial toxin. There are two of them, alpha toxin and the enterotoxin. Okay. First, an overview of the Clostridium species. All of them are gram-positive bacilli. They are all obligate anaerobe. That means they die when they're exposed to oxygen. Okay? The reason they die when exposed to oxygen, this is true for all anaerobes, is that they cannot detoxify. Okay, oxygen metabolism always leads to hydrogen peroxide and hydroxyl radicals. Aerobic bacteria are able to detoxify using specific enzyme called catalase and superoxide dismutase. These enzymes are totally missing in anaerobe. So when exposed to oxygen, when H2O2 and OH are produced, these cells simply die because these are two extremely toxic oxygen species. Okay. In any event, these are obligate anaerobe. There are three major clinical species: the tetani, botulinum, and perfringens. Okay? Now, once upon a time, Clostridium difficile was also assigned to this same genus. However, in 2016, it was moved to its own genus called Clostridioides. This is the currently correct name, Clostridioides difficile. We're not going to deal with this because we already dealt with it before. All right, let's go to tetani. First, look at the organism. It's a gram-positive slender rod, and it has a characteristic. It has terminal spores that gives a very characteristic drumstick appearance. You can see that very clearly in this stain image. Drumstick-like morphology, characteristic of the tetanus bacterium. It is distributed in feces and contaminated soil at very high density. The infectious form is a spore, and it can only grow in an anaerobic environment. In the United States, there are a small number of deaths, 50 to 70 in the United States, but worldwide the number is large, 1 million deaths due to tetanus worldwide. The pathology of tetanus is spastic paralysis, shown in this young patient whose arms are contracted and who shows rigidity of the neck and the jaw and so on. And in the depiction of a 19th century painting, a patient suffering spastic paralysis captured well in this painting. Okay? The diagnosis is based on clinical symptoms, and often there is no infection because this is an intoxication. There's only a small number of bacteria. It's mostly based on clinical symptoms. So what in the world is tetanus neurotoxin, and what does it do? And how does it produce the characteristics of the tetanus disease? It's a simple AB toxin. The activity of the A subunit is the endopeptidase. The B is the receptor binding domain. It also has a translocase domain. It is shown diagrammatically here. The A subunit is a zinc-dependent protease, and the B subunit, which in turn consists of a binding domain and a translocase domain. So this is a classic AB toxin. So where does the tetanus neurotoxin act? Obviously, it acts on the central nervous system, but where exactly does it act? It turns out that toxin binds to neuromuscular junction. It doesn't bind to the brain, it binds to the neuromuscular junction, shown diagrammatically here. Nerve ending and muscle, right? The neuromuscular junction. However, this is very important. It does not act there. Instead, the toxin is transported via the axon to the next synaptic junction with an inhibitory neuron. So the toxin is absorbed with the neuromuscular junction, but transported axonally and delivered to a synapses with an inhibitory neuron. What's an inhibitory neuron? In normal control of a muscle, the brain sends signals to contract a muscle, but once the job is done, it has to make the muscle stop contracting. So to make it stop contracting, it has to send an inhibitory signal as well. That inhibitory signal is delivered at the synapses between an inhibitory neuron and the neuron that actually lead to muscle contraction, right? So the toxin reaches ultimately the inhibitory neuron and it exerts its action in the inhibitory neuron. The activity of the toxin in the inhibitory neuron is such as to proteolytically destroy a specific protein called synaptobrevin. What synaptobrevin is actually made more clear in a subsequent slide when we're talking about botulinum toxin. But basically, for our purposes here, it acts in the inhibitory neuron. It destroys a particular protein which prevents the inhibitory neuron from inhibiting the contraction. As a result, the inhibitory neurotransmitter called gamma aminobutyric acid or GABA is not released into this neuron. As a result, the muscle is in a state of continuous contraction. It cannot stop contracting because the inhibitory signal is not being delivered to that neuron. This is what causes that characteristic spasm associated with tetanus. That is, continuous muscle contraction without any inhibition of that contraction. Okay? All right. So here is more information on the disease caused by Clostridium tetani. How do you treat patients with tetanus? One of the things you do is to isolate the patient away from stimuli because stimuli cause excitement and excitement lead to muscle contraction and muscle contraction which cannot be inhibited because the presence of the toxin will exacerbate the disease further. So the isolating them from stimuli is a good start. And the treatment is from anti-tetanus immunoglobulins. And if the tetanus infection has started at the surface, then debriding the wound, that is surgically opening up the area and scraping off the tissues that are infected along with the bacteria is very important. An antibiotic to prevent further growth is also important. Tetanus is basically prevented by vaccine again, the same vaccine. But the vaccine needs to be repeated every 10 years, otherwise it loses potency. Okay, now we'll move on to Clostridium botulinum is a gram-positive slender rod. It also has terminal spores, but it's not quite drumstick-like, but it does have terminal spores. Okay? Botulinum is named after botulus, which means sausage. There are about 40 to 80 cases per year in the United States, more worldwide. The pathology of botulism is flaccid paralysis. Botulism is characterized by many symptoms, neurological symptoms, including slurred speech, double vision, and difficulty swallowing, and progressive flaccid paralysis leading to arrest of breathing, there is respiratory paralysis, and in cardiac arrest. Okay? There are three types of botulism. The most common is the classic botulism, which is a foodborne intoxication. This means preformed toxin that is present in the food is ingested that leads to this intoxication. The second kind is growth in wounds, which is very rare, quite rare. And the third kind, also quite rare, is something that happens only in babies. There's bacterial growth in their intestine. These bacteria are often acquired from honey. That's why you get the admonition never to give honey to babies. Both of these kind, wound kind and infant kind, are quite rare, much more common in the foodborne intoxication. Okay, so the same information is given here, three different forms. The bottom line is whether it is foodborne or infant or wound, ultimately the toxin reaches the bloodstream. By the way, the toxin, which is a very large molecule, is nevertheless absorbed quite effectively in the intestinal epithelium because it targets a specific epithelial cell, which internalizes the toxin and delivers it into the interior, ultimately into the bloodstream. It is delivered by bloodstream into the neurons, where it leads to flaccid paralysis. So what exactly is flaccid paralysis and what is the mechanism of action of neurotoxin? That is shown in this slide. The botulinum neurotoxin is also a simple A-B type of toxin. It has an A domain, which is a peptidase or a protease, and it has a B domain, which in turn consists of receptor binding domain and a translocation domain. They're held together by a disulfide bond, just as in any other type of AB toxin. Okay? In order to understand what botulinum neurotoxin does, how it brings about flaccid paralysis, you have to look at how a normal nerve stimulation of muscle works, and then understand how that normal nerve stimulation of muscle is compromised in the presence of botulinum neurotoxin. The normal situation is shown in the upper part of the diagram. What is shown in the inset here is a neuromuscular junction. Here's the neuron, here's the axon terminal, and here's the muscle cell. It is shown in amplified state here. This part of the diagram is the axon terminal. This part is the muscle, and the space in between is the so-called synaptic cleft. And this light colored membrane here, which is part of the axon terminal, is the synaptic membrane. Okay? In normal muscle function, normal nerve stimulation of muscle, a small chemical, small molecule called acetylcholine is released into the synaptic cleft, where it is taken up by the muscle cell, and that acts as a signal for the muscle to contract. So in order to make a muscle contract, the axon has to deliver acetylcholine to the synaptic cleft, where it is taken up by the muscle cell, and that leads to contraction. This is the normal process. Delivery of acetylcholine requires the following steps. Acetylcholine is not present in free form inside the axon terminal. Instead, it is enclosed in vesicles called synaptic vesicles. So the small molecule that's confined to membrane sacs called vesicles, and these vesicles need to be delivered to the synaptic membrane, where their own envelope needs to fuse with the synaptic membrane so that their interior can be emptied out into the synaptic cleft. So this is the mechanism for delivering acetylcholine to the muscle cell. That is, synaptic vesicles need to fuse to the synaptic membrane, and then only then acetylcholine can be delivered to the muscle cell, right? This is the normal process. What happens? So, in order for this vesicle to reach the synaptic membrane and fuse, this process is called docking, membrane docking, and vesicle fusion. This requires proteins that are together called snare proteins. Those are the proteins shown in wiggly, colorful lines here, blue, red, and yellow. These are snare protein. One member of the snare protein is called synaptobrevin. Okay? You encountered synaptobrevin in the context of tetanus previously. The same synaptobrevin is also targeted by botulinum neurotoxin. What happens in the case of botulinum neurotoxin is shown in the bottom part of the diagram. When the botulinum neurotoxin is present, then that AB toxin is taken up at the neuromuscular junction, and the active domain, the A subunit, cleaves synaptobrevin such as to destroy the snare protein complex that's required for vesicle fusion to synaptic membrane, therefore, which ultimately leads to blocking of delivery of acetylcholine to the muscle. So the muscle never contracts. Okay? So what neurotoxin, what botulinum neurotoxin does is it blocks release of acetylcholine, which is necessary for muscle contraction. So here is a trick then. Both tetanus toxin and botulinum toxin target the same exact protein, synaptobrevin. However, they have completely opposite consequences. Tetanus toxin leads to continuous muscle contraction. Botulinum toxin leads to no contraction at all. How is that possible? It's possible because of where exactly the synaptobrevin is cleaved and what the consequences of that synaptobrevin cleavage in that particular context is. In the case of botulinum, the toxin is taken up of the neuromuscular junction and it acts at the neuromuscular junction. Its action prevents acetylcholine release, therefore, it blocks muscle contraction. That's clear, right? So, how is that different from tetanus toxin? Well, it turns out that in the case of tetanus toxin, which is shown here, the toxin is actually, tetanus toxin is taken up at the same neuromuscular junction, but it does not act there. It does not act there. Instead, it gets transported through the axon and is delivered to the next synapses with the inhibitory neuron. And it acts at the axon terminal of the inhibitory neuron. There it cuts the synaptobrevin, which prevents the inhibitory signal, GABA, not acetylcholine, but GABA, from reaching the neuron. That means no inhibitory signal is being delivered to neuron, but normal acetylcholine is being delivered here. Here's the normal process. So in this case, the muscle is in a continuous state of contraction because no inhibitory signal is reaching the neuron. Whereas in the case of botulinum toxin, both the uptake and the activity are at the same neuromuscular junction. Here, the cleaving of synaptobrevin lead to a blockage of acetylcholine delivery to the muscle, and therefore there's no contraction occurring. So you have a paralysis because muscles are not contracting. Good. All right. So Clostridium botulinum treatment. The treatment requires an anti-botulinum antitoxin because this is an intoxication, right? In most cases, botulinum intoxication is because of injection of the toxin, not the bacterium itself. And in the case of the rare cases of antibiotic, rare case of infant or wound botulism, you do use antibiotic to control the infection. Prevention of botulinum intoxication by proper food handling. Okay. Now, botulinum toxin, about 4 decades ago, was approved as a treatment initially for medical conditions, but later it became commonly used as a cosmetic. Originally, it was approved for treating specific medical conditions. For example, facial paralysis. These patients, when given extremely low doses of botulinum toxin, were helped. As you could see in this patient, the symptoms of facial paralysis were alleviated significantly by using very, very low doses of botulinum toxin. Incidentally, botulinum toxin is effective for up to 3 to 6 months at a time. Then it will have to be repeated in order to regain the same effect. It is also used for strabismus, seen in this patient, and in this case of the patient also there is significant improvement in in alleviating the symptom of strabismus. But in subsequent decades, it was also begun, because it's approved, it's also been used as a cosmetic, for example, in wrinkle mitigation, as shown in this case. Right? So Botox, as it's called, Botox is the brand name, basically is botulinum toxin in extremely small doses. Okay, now we go to the third Clostridial species, Clostridium perfringens. This is a gram-positive bacillus. It also forms spores. It's also an obligate anaerobe. It resides in vertebrate gastrointestinal tract, but also in it's also in soil, and it has its spores are present in the soil. Okay? Its pathology. Its major pathology is gas gangrene, also known as myonecrosis. This is it's a pretty serious condition. It also causes a separate pathology is food poisoning. In fact, 7% of food poisoning in the United States is because of Clostridium perfringens. The virulence of this species because of two different toxins, alpha toxin, which is responsible for gas gangrene, and Clostridium perfringens enterotoxin, which is responsible for food poisoning. Both of these, neither of these is an AB toxin. Both of these are actually membrane disrupting kind of toxin, but they work by two different mechanisms, as we will see. Alpha toxin is a phospholipase. What it means is it destroys the cell membrane of the host target cell by cutting at a specific chemical entity called lecithin or phos. Okay? So in this case, the toxin has two domains. One domain binds to the cell membrane. The other domain basically destroys the membrane locally. And this hydrolysis leads to membrane disruption, and that leads to a very serious condition called gas gangrene that starts an opportunistic infection by not only Clostridium perfringens, but also other bacteria that invade the cells that have been permeabilized. Right? So this causes an opportunistic infection, and usually Clostridium perfringens infection starts at sites of a severe wound, such as battlefield wounds, surgical wounds, and also venomous snake bites. Now, venomous snakes have mechanisms for injecting venom into their prey, and the mechanism for injecting the prey is by inflicting a deep wound into the tissues of the prey, right? So that same wound also creates conditions for Clostridium perfringens infection. So the spores that are delivered that way grow rapidly. in anaerobic conditions, and that leads to eventually an abscess formation, and the gas is formed because anaerobic bacteria are fermenters, right? They can only extract energy by fermentation, by fermenting tissue carbohydrates, and they form abscesses. That means a lot of pus, and they produce foul-smelling gas. So, and a black pigmentation. Okay? Usually, gas gangrene is very bad news. Gas gangrene is usually fatal. Okay? All right. So the other toxin, which also acts on the membrane, is the pore-forming enterotoxin. These pores basically, these proteins basically embed themselves in the host cell membranes. In this case, the epithelial cells in the intestinal tract, and they trigger apoptosis by the usual mechanism, which is a release of ions, an influx of water, and so on and so forth. And this leads to diarrhea and other GI symptoms. As we mentioned, 7% of food poisoning is because of Clostridium perfringens, pore-forming enterotoxin. Okay. Diagnosis of Clostridium perfringens infection is mostly on clinical grounds. The patients display disproportionate local pain. They suffer from tachycardia, tachypnea. There is rapid heartbeat and rapid breathing, fever, delirium. The skin becomes stretched, necrotic, blisters, blisters are seen, and then there's black pigmentation and malodorous discharge. The treatment is almost invariably there is surgical debridement shown in the diagram. That is, that you scrape off the tissues that are infected. Penicillin 4 is given to control the infection, and in recent years, hyperbaric oxygen has been given, but this treatment remains controversial. Hyperbaric oxygen means putting them in an oxygen tent and increasing the oxygen pressure so that they are oxygenated. The idea here is that Clostridium is an anaerobic organism. In the presence of high levels of oxygen, it will get killed. But how effective it is controversial, but it has been practiced. Okay, that's the last slide. That concludes our lecture. Thank you very much. I apologize for going over time by 7 minutes.
Thank you. Thank you.
Okay, let me see if there are any questions. Okay, there are no questions that I see in the chat.
All right.
Okay, I'll stop the recording then.
Yes. Thanks, Afri. Thank you very much.
Thank you, Ian.
Take care.
See you in another few weeks. Bye-bye, everyone.